What's good, everyone? Welcome back to Meowba Academy. And today, my topic of interest is going to be glucogenesis. Now, you might think I've talked about this before, but you would be confusing this term with gluconeogenesis. This is a bit opposite. Well, this is the formation of glycogen with the presence of excess glucose in our body. So, when there's a large amount of glucose in our body, it is converted into glycogen because a large amount of glucose can cause osmotic imbalance. Therefore, it can be stored just as glucose in our body. So, to maintain the osmotic balance in our body, it is converted into glycogen. Whereas the gluconeogenesis I talked about before was the formation of glucose from non-carbohydrate products. So this is the opposite. This is the breakdown of this is the polymerization essentially of glucose into glycogen. Well, this occurs in liver and muscles, and it is composed of three important steps, and I'll be writing them. So these are the three steps. I will be explaining them further. So at first, the glucose binds with ADP to form UDP glucose. Glucose binds with UDP with the help of special enzymes, and then the glucose binds with glucogenin to form 4 carbon chains, and then the proper formation of chains and branch starts. So, the first step, as we talked about, is done with the help of an enzyme called phosphoglucomutase. What this essentially does is that it converts glucose 6 phosphate into glucose 1 phosphate, which further releases UTP. So this is the energy that is released by the movement of a phosphate bond from 6 carbon to first carbon. So this energy is released with the help of phosphoglucomutase. And then with the help of the enzyme UDP glucose pyrophosphorylase, that UDP is converted into UMP. So this is uridyl triphosphate. It is converted to uridyl monophosphate, essentially releasing 2 molecules of phosphate. And that one molecule of UMP, it binds with this glucose 1 phosphate. And you would assume, like, you would wonder how how UDP glucose forms, because this is UMP, this is not UDP. So what happens is that one phosphate from UMP and the other phosphate from glucose 1 phosphate bind together to form 2 phosphates. So therefore, UDP glucose is formed. So essentially, this reaction keeps happening again and again until there is enough UDP glucose to form glycogen, because glycogen is a polymerized form of glucose, and we would need a lot of glucose to form that, and then the glucogenesis starts. So in the second step, we would need to form 4 carbon chain of glucose, so that it would be recognized by enzymes to be considered a molecule of glycogen, because if it's shorter than 4 carbon, our enzymes wouldn't notice that it's actually glycogen, not glucose. So with the help of the enzyme glycogen synthase, our enzyme um, glycogen synthase uses what it does is that it helps this protein glycogenin bind with the glucose UDP glucose molecules to form a 4 molecule chain. So now that we have a 4 molecule chain of glucose, it then binds with UDP glucose. Now that it binds at a special place and makes a bond called alpha 1,4 glycosidic bond. It binds at this position. So this is the chain formation. I forgot to mention. A chain is formed with 4 molecules of glucose and 1 molecule of UDP glucose, and it keeps happening until a chain is formed, like so, at alpha 1,4 chain. And lastly, another enzyme is used, which is called the branching enzyme. What it does is that it converts the chain into branches, because we just can't have long chains. The chains are like 6 to 8 molecules long. So a branching enzyme is used to make branches at alpha 1,6 glycosidic bond. And that's it. That's all it takes to form glycogen from glucose. And if you have any other questions or any other constructive criticism, do let me know. And make sure you like and subscribe. That would help a lot. Thank you.